Hello, everyone, and welcome to our second webinar of our first series of Agriculture and Biotech, which are brought to you by Food for Sustainability Academy and the Grow Partners. My name is Claudia Costa. I'm the academic director of the Academy of Food for Sustainability. And in today's session, which is titled Smart Tools for Soil Decision, we will look at how farmers and agronomists can move from this gut feeling to something that is data-driven soil management by using complementary tools such as barcoding for soil microbiome, honey-type holistic chemistry, and enzymatic activity assays to support practical decisions in the fields. So I really hope that you enjoyed our session today and that you feel encouraged to participate in the discussion. And let me just very briefly remind you that Food for Sustainability, our soil health and function laboratory, was created precisely to make this type of information accessible and actionable. So through molecular methods like metabar coding, we profile bacterial and fungi communities, and with chemical and emerging enzymatic analysis, we can capture how these communities are functioning in terms of nutrient cycling and, of course, soil resilience. And altogether, these tools help translate complex soil biology into clear recommendations about fertilization, organic matter management, and risk of degradation. And so we don't leave these insights locked in the lab. They drive real world impact that positions food for sustainability at the forefront of translating soil science into resilient farming systems. So let's take a few more examples. We have here Ampliaqua, which is our multi-trophic system that combines microalgae, fish, and vegetables. And here, microbiome and water quality data guide how we design bio-based inputs and also how we can close these nutrient loops, proving that in practice, smart measures can actually slash losses and synthetic inputs and very important risks. And we also have a flagship project called Idanha Rural Food Park, where soil and pasture diagnostics will underpin regenerative protocols across extensive and silvopastoral systems, which directly link soil function to superior meat quality, carbon storage, and lasting territorial value. And this expertise from lab to commercial scale is exactly what our outstanding speakers will unpack today for us to join the discussion. I'm very pleased to have with us Joana Cruz, Soil Health and Function Laboratory Management at Food for Sustainability. She specializes in plant-associated microbial communities and sustainable soil management. And Joanna will introduce how metabar coding and microbiome interactions can be integrated into farm diagnostics. We are also have the pleasure to be joined by Alan Evans. Alan is the technical director at A2 Chemical Analysis. Alan has deep expertise in agricultural laboratory analysis and sustainable agronomy and will walk us through holistic tools such as the honey test and how to read them from the farmer's perspective. And finally, but definitely not least, we are joined by Ruth Pereira. Ruth, she's associated professor at the Science Faculty of University of Porto. She's also the director of GreenUPorto and head of the LABRISK. And Ruth's work in soil and water risk assessment and environmental ecotoxicology will help us understand how enzymatic activity and other biological markers signal risk and hopefully also opportunities in our soils. So it's a big, big thank you to all the speakers. But before we dive in, let me quickly set a few house rules so to ensure that everything runs smoothly for us. So the webinar will wrap up at half past the hour sharp. After each speaker has his presentation, we will open the chat for Q&A answers, Q&A. So that's where your questions should go, and I will direct them to the speaker. The session is being recorded, so please feel free to switch off your camera if you do not wish to appear, but I truly encourage you to keep your cameras on. It makes the experience much, much nicer for everyone, and especially for our speakers. Also remind you that at the end, you will get your certificate once you complete a very short quiz. Super, super quick. I promise it's going to be quick and easy. And again, a reminder that we have some prizes for those that complete the entire sessions of the quizzes. Okay? So we are about to start, but let's do a warm up. It's, very cold outside. So can we all go to Slido and answer a couple of questions around our topic? Let's, get really immersed into the topic. So I can see that we have Slido on. So either you can use the QR code or simply use your browser and just type www.slido.com in the hashtag box. Just insert F for F Academy. Let's jump into Slido. Everyone into Slido. Okay. We have a couple of questions here for you. Are we ready for the first question? Are we? Sophia, you're smiling, but I cannot see the question. Thank you. And the first question is great. I think this is the second question. Oh, is it the first one, Sophia? This one? Okay, so which type of smart tool is the most used to assess soil microbiome at scale in Europe? So we have drones, remote sensing, high throughput DNA sequencing, so matter barcoding, soil sensors, satellite images. So smart tools to assess soil microbiome at scale in Europe. Okay, so you are voting. Okay, it seems like we have a clear winner. No, so sensors are also an option. But let's consider that soil microbes are invisible but crucial, of course. DNA sequencing reveals them. So matter barcoding captures microbial diversity by extracting DNA from soil and sequencing barcodes. You know, the 16X for bacteria and the ITS for fungi. So it can identify thousands of species at once, including uncculturable microbes. Remote sensing, drones, satellites, they detect surface patterns or moisture, but they cannot see the microbes themselves. So matter barcoding provides a direct view, and as such is our correct answer. Well done, guys. Very good. So matter barcoding is the one that is the smart tool used to assess soil microbiome. Good. Feeling warmer, everyone? Ready for the second question? So which of statements about soil tests is true? So we're getting a little bit more into the topic. Matter barcoding tells you how fast soil enzymes work, how fast. Enzymatic activities tests, they actually show how active soil biology is, namely in cycling nutrients. Honey test It identifies the fungi and bacteria that are present through their DNA. Or is it that all three tests measure exactly the same thing? So matter by code, how fast enzymes work, enzymatic activity, how active soil biology, the honey test, knowing how which fungi and bacteria are present, or are they all the same? So let's see. Matter barcoding tells you which microorganisms are present by reading their DNA barcodes. So it's like taking a census of who's living in the soil. Okay? The honey test pulls together multiple chemical and biological measurements, and Alan will tell us about things like respiration rates, organic carbon, and some other. And then it puts all this into a soil health score, so not focusing in single enzymes. And enzymatic activity tests, they measure how quickly specific soil enzymes like the ones, and Ruth will tell us a little bit more about this, handling carbon, nitrogen, or phosphorus, are working right now, making them a very good proxy of how biological, active, and functional our soil is, which makes the first question, the first answer, the correct answer. Well done. And this was no question about which of these sentence was true. Very good. Feeling warmer now? Do we have one more question? Yes? No? We do have one more question. So, which of these approaches have been used in soil science for the longest? Okay. Matter barcoding, enzymatic activity, honey test, AI modeling of microbial networks. Which one has been around for longer? So a little bit of explanations here before you show. Let's let everyone uh, take a moment to think about. So the honey test is actually the newest approach, and it's a comprehensive soil health index that integrates chemical and biological indicators, specifically designed for farmers. Okay, so not a honey test. Matter barcoding profiles microbial DNA communities, and it really took off with the next generation sequencing around a decade ago. Enzymatic activity. It actually builds on classical soil biochemistry research and has been around for several decades. So the correct answer, and congratulations those that answer, enzymatic activity. Indeed, been around for several decades. This makes them the oldest of the three approaches in terms of soil science. Okay, so thank you so much for answering. I think we now feel warmer, right? Thank you so much for to the team to having put these questions for you all. So let's now dive into how microbes insights and smart soil tests can become everyday decision tools on the farm. Alan, I see that you are ready. So we will start with Alan. I'll call you to the floor. Thank you.
Thank you very much. Sorry about this little problem here. Good afternoon. My name's Alan Evans, and as you heard, I'm the technical director of a Portuguese soil analysis laboratory called A2 Análises Químicas, which means chemical analysis in Portugal. It's just a very quick introduction to our laboratory. As you can see, we're based in northern Portugal, about 1 hour or so away from the Douro wine country, which most of you have heard of. And we were also in the heart of a place called Minho, which is where we can get lots of lovely vinho verde or green wine. So our laboratory is set up to analyze all types of things for agriculture: soils, leaves and sap, organic amendments, fertilizers, substrates and waters. We don't carry out that much microbiological analysis here. We're more based on chemistry, but we do have microbiological services through our partners. Now, when the lab started about 2007, we were carrying out the classic chemical tests for soil analysis. But from about 2023, the regenerative agriculture movement and the soil analyses necessary for regenerative agriculture really started to kick off. So we started to pivot a little bit to that. And what I'm going to talk to you about today is the Haney test. So first of all, we'll have a quick look just to see how the Haney test does differ from the other types of tests that we have. Let's just get this on. So a classical soil analysis test, I've based it on Portugal, but it's pretty much the same more or less throughout the world, where we'd be getting a pH, conductivity, lime requirement if necessary, texture, total organic carbon, organic matter, all these things you can see here. MELIC3 extractable elements. Of course, there are many different ways to extract the elements and nutrients from the soil. MELIC3 is the one that we normally use here in Portugal. Cation exchange capacity, we also have various methods to use that. Our preferred method here in our laboratory is the cobalt hexamine method. But if we take a look at all these, what we see is that these are all purely chemical analyses for the soil. There's no real biological parameters in there. So what happens here normally is you'll get chemical analysis, and then you'll get in return a chemical recommendation. What I mean by that is that you'll see, okay, your soil analysis says that your phosphorus values are low. So here you should be applying this amount of phosphorus to the soil to put the nutrients up to adequate levels. And the same thing happens with all other nutrients. What is not happening here is we're not trying to get the microbiome working and releasing nutrients for itself. However, the Haney test does actually include some biological indicators in the analysis suite. We see we have the classic pH and electrical conductivity, organic carbon, organic matter. But here, things start to get a little bit different. We now have water extractable organic carbon, something called the soil CO2 burst, percent MAC, percent MAC, percent microbial active carbon. Now, these tests are relatively new for, let's say, mass-produced soil analyses, not necessarily things to do with education or maybe research projects. And the interesting part here is that these are all to do with microbial, let's say, chemical microbial characteristics of the soil. So water extractable organic carbon, soil CO2 burst, and the percent microbially active carbon are actually some biological indicators that we can use to take some steps to improve our farming procedures. Here we also have a few things that are different. Nitrate and ammonium, okay, we all probably do that anyway, routinely. But then we now have water extractable nitrogen, which is the total nitrogen from the soil, and the water extractable organic nitrogen. So a little bit different there as well. And these, once again, microbial parameters, which can help us. Then we have something here different from MELIC3 extractable elements, something called H3A-4. That's because it's version 4 of it. These extractable elements, and we can also get something from these tests, which is the organic phosphorus. One other important difference here that the Haney test provides is this soil health score, and we'll talk about that in a little bit. So let's just have a look at a few things here. 10-15 minutes probably isn't enough time to talk about everything that we need to talk about here, but just to give you kind of a flavour of what the Haney test is, what it can do, and the type of information we can get from it. So we talked about the H3A4 extraction. H3A is actually not a chemical compound or anything like that. It's just the names or the initials of the names of the people who invented this extraction solution. off the top of my head, I'm not sure they are. Haney is definitely one of them. So that's one of the H's. Then there are two more H's and an A somewhere in the middle. Now, if you look here, we've got the reagents that are used and the concentration. And what we see is that these are all what we call low molecular weight organic acids, which are normally things that are produced either root exudates of the plants or actually produced by bacteria and fungi in the soil. And they are more or less in the concentrations that we'd kind of expect them to find in the soil. So what we're using here with this H3A4 extraction are actually chemicals that we can find in the soil and that could be used by the plants, the microbes, to release nutrients from their surroundings. This is a little bit different to, let's say, melic 3, for instance, which in its chemical composition has nitric acid, a concentrated acid. It has ammonium fluoride, for instance. Somebody maybe will correct me if I'm wrong, but I don't think there are any animals or bacteria that actively produce ammonium fluoride. Now, if you remember, the ammonium fluoride that we use in the melic 3 soil test is there to release phosphorus from the soil. That's its main purpose of being there. But in the real world, in nature itself, there are no animals, bacteria, plants that will actively release fluoride into their environment to dissolve the phosphates in the soil. So what we see here, we've got these low molecular weight organic acids, which do have a very significant role in the productivity of the soil. And for those who are more chemistry based, these are all something we call carboxylic acids. They have a carboxylic acid group, which is a C-double-OH. And there are different types of them, monocarboxylic acids, dicarboxylic acids, and tricarboxylic acids. Here we have citric acids, a tricarboxylic acid, oxalic and malic acids are dicarboxylic acids. These will extract all the nutrients, the key from the soil. One thing you might notice that is missing is boron. We cannot extract boron using the H3A extraction reagent because of a chemical reaction that occurs with citric acid. And it is possible to distinguish between extractable organic and extractable inorganic phosphorus in this method. Here are they distinguished between ICP, the analytical technique we use to do the total phosphorus, and ultraviolet visible, which we use to get the phosphate. We also have, for the Haney test, as we saw, water extractable organic carbon, water extractable nitrogen, and water extractable organic nitrogen. The importance of these, like I said, these are to do with the kind of microbiological chemical parameters of the soil. This will give us an idea of how much food, how much fuel is there for the soil microorganisms. So high values of water extractable organic carbon, water extractable organic nitrogen. So things like amino acids, proteins, sugars, all these type of things that the microorganisms and plants can use. We'll be quantifying them there in this test. Okay, and we'll be able to see how much is in there and get an idea of whether it's going to be good for sustaining the microbiological life in the soil or not. Then in conjunction, we have this other test, which is the soil CO2 burst test, which is going to give us a general idea of the amount of microorganisms in the soil. It's not going to be identifying or anything like that, like no meta barcoding or anything like that. We're just going to say there are this amount or this number of microorganisms in the soil. These microorganisms produce carbon dioxide, and we measure the carbon dioxide they produce. It doesn't tell us anything about what the microorganisms are, whether bacteria, fungi, or anything else. So what we do in this test is we have air-dried samples, we re-wet them, and what happens when we re-wet them is that the microbes in the soil receive the water, become hydrated once again, and start carrying out their life cycles. As they carry out their life cycles, they respire and release carbon dioxide. And this carbon dioxide is measured here on this little paddle thing. As you can see, we've got blues, yellows, greens. The more carbon dioxide that's released in a 24-hour period, the more yellow that this paddle here will become. So if we see here, this soil here has a very low number of microorganisms in the soil because it's producing a very low amount of CO2. This one here, which is more yellow, has a higher amount of microorganisms in the soil. So as we see here, the more CO2 released, the more microbes are present in the soil. Once again, we cannot identify them or anything like that. What I've got now is just a few, because I don't really have that much time. I'd love to go into more detail about lots of different things. Just a few of the analyses that we've got in Portugal of these varying tests from the past 3 or 4 years that we've been carrying them out. So water extractable organic carbon. So we see here we have the levels. So any or extractable organic carbon less than 100 milligrams per kilogram is considered low. Then we have average and high. Now, this is water extractable. Please don't confuse it with the total organic carbon, which we are probably more accustomed to seeing on the soil analysis report. Total organic carbon and organic matter. This water extractable carbon is only a very tiny fraction of that. As you can see, we have these values here in milligrams per kilogram, whereas organic carbon is normally in a percent value. And as probably would expect here in Portugal, we've got kind of average values here for water extractable organic carbon. And these normally here, the bulk of these farms here are ones who are just starting the journey in regenerative agriculture. So this is really kind of like the baseline for Portugal. We did have a few farms here which had very high water extractable organic carbon values, but these were actually farms who were already had their regenerative farming setups in good working order. Water extractable organic nitrogen, I think, is a little bit more interesting here in Portugal. As you can see, even the people who had very well set up regenerative agriculture farms, the amount of water extractable organic nitrogen in there was still quite low. And if I'm being honest here, I probably could have divided this graph up into another bar chart, so have one more section. Because most of these values are between probably about 5 and 30. There are only very few here that were above 30 milligrams per kilogram of water extractable organic nitrogen. And last slide, just because I think my time's running out. Here are some soil CO2 burst results from Portugal. So once again, we've got the levels here that we consider. So less than 6 milligrams of carbon in the form of carbon dioxide that's produced. We have very low microbial activity, very low biological activity. Then we go low, average, high, up to high here. In Portugal here, most of them here are situated in this zone here. So in the very low region here. So really, the farmers here in Portugal are going to have to probably put in quite a bit of work to increase the microbial mass in their soils, because then this microbial mass can then be used instead of putting all the nutrients in the form of mineral fertilizers. We can actually use the microbiome to be destroying the soil, destroying in inverted commas, to be releasing these nutrients from the soil, which are already present in the soil. And that's kind of the advantage of the Haney test, is that we have some microbiological parameters that we can use to help make decisions as to what we should do, if necessary, to increase the microbial communities in our soil and to use them instead of mineral fertilizers. Thank you very much for your attention.
Thank you very much, Alan, and thanks for being so strict about the time, because our idea is to actually give time to our participants to raise some questions. I'm sure that there will be questions. Just let us give some time to the audience. Alan, if I may start with a clarification question. So Metavar coding give us this very high precision by reading DNA to identify exactly which bacteria and fungi species are present and their proportions. So how do you see it complementing the honey test with the snapshot of the overall soil biology? And I can go just a little bit further. say that we take, for example, fruits and vegetables, and we know that Pseudomonas populations suppress specific pathogens, or that Trichoderma boosts some root health, and we could have, specific recommendations like, oh, just add some nitrogen fixing inoculum, versus the honey test that give us something a little bit broader, something like, just add more organic matter. So what are your thoughts on combining both approach for something that is actually actionable for
Hmm.
farmers?
Yeah, so let's just start by thinking about nitrogen then. So as we saw in the graphs for Portugal, the amount of organic nitrogen in the soil was generally quite low, average to low. And now that's probably not because of the lack of organic matter in the soil. As I said, 10, 15 minutes isn't really a lot of time to go into depth of these subjects. Um, so really, the soils in Portugal, most of them have a reasonable amount of organic matter, up to medium values, and the amount of nitrogen contained in there is actually quite a good amount. So what we could actually use this test for, or the barcoding and the DNA analysis, is actually see if we do have the necessary types of bacteria in the soil that could actually degrade the organic matter, change it into smaller, more bite-sized, shall we say, fractions of organic matter, which the microbes can then use use to complete their life cycles, use that as an actual food. Because at the minute, what normally happens is we're throwing in ammonium and nitrate into the soils to give the plants and indirectly the microorganisms a food source. And with regenerative agriculture, we probably want to try and shift away from that a little bit. So the Haney test can give you an indication of what's in the soil at the moment, the amount of sugars, for instance, from the water extractable organic carbon. We have high amounts of water extractable organic carbon. Then we can say, okay, the microorganisms do in fact have a good food source there. Um, when we see low values of water extractable organic carbon, what we can actually see is that the microorganisms perhaps don't have enough food for them to just carry out their most basic tasks, shall we say. So we need to increase the amount of water extractable organic carbon in the soil to make sure that we were giving the microorganisms the food and the fuel that they need to carry out the tasks that they need to do, let's say, on a day-to-day basis. So
Okay.
the barcoding can probably help identify which types of bacteria and fungi that we do have in the soil, and then say, well, okay, we do have these in there that will be breaking down like the carbon-carbon bonds, like the glucosidase enzymes, and some other enzymes that might break up the nitrogen. And then if we can get them working, reproducing, increase their populations, then we can turn the soil organic matter that's there into an available food source for them.
Okay. Thank you. Thank you so much, Alan. Just one other question.
Mhm.
So we just saw from our warm up that this is a fairly new test. It's a more recent
Mhm.
test, and but we are talking, you know, other concepts, other ideas. So in terms of the chemistry and the biology of the soil, and you also showed that you have been sampling Portugal, and so what is your response to how all this information is being picked up by farmers? Is
Mhm.
it still far too complex? I don't know about the prices. Is it still
Mhm.
expensive? So in terms of, disseminating this knowledge, so what is your view
Mhm.
in terms of uptake and what we need to do to promote it further?
Okay, so the analysis itself is not that much more expensive than a normal soil analysis test. It's around about 50 euros is what we're talking here, more or less. Um, the difficulty really in Portugal is the interpretation of the analysis reports, because when it's a classical analysis report, they can look and say, okay, I need to put more phosphorus in here. So let's put 200 units of phosphorus in the soil, and I've solved my problem with low phosphorus. However, with the Haney test, the idea is to try and reduce the amount of fertilizer used. Obviously, it isn't something you can do one day to the next. It's a process that's going to take a few years. Reduce the reliance of the plants and the microbes, for that matter, on fertilizer, mineral fertilizer inputs, and try and transition it so that the microbes themselves are breaking down the soil particles, breaking down the organic matter, and releasing the nutrients to the plants. So the Haney test is kind of a way to do that, but because it's a recent thing, the agricultural technicians and the advisory boards are not really 100% up to date on how to use that information, which is one of the challenges we found in trying to get people to take up the Haney test. It's really disseminating the knowledge to the farmers themselves.
Okay, thank you. But Alan, you pointed something also very important is that this transition takes time. And let's not be radical about taking out all the inputs into the systems, particularly in systems that are very dependent on these inputs. It's also a very important idea, and we do have tools to assist that. Just two clarification questions. Alan, I will start with the last one. If the dissolved organic carbon is different from the water extractable organic carbon, are they different?
The dissolved organic carbon is probably not different. I think we're just talking about different terminologies here. Water extractable is where we'd actually take a soil sample, add water to it to remove like the organic carbon. Dissolved organic carbon could be just in the soil water itself. So it's not an extraction procedure per se. Probably what I'm thinking is more just semantics, but it's more or less the same thing.
Okay, thank you. So, and one final, this is a more broad open question. So, in your opinion, how could the bacteria and fungi identification impact the climate change, the impact that also climate change has on soil quality in relation to sustainable soil management?
So what we're trying to do really with the Haney test and these biological analyses in themselves is we're trying to see or trying to profit from the microorganisms that are in the soil that for hundreds, thousands of years have been in the soil and working, destroying, recycling nutrients, and then allowing the plants to grow. So the idea here would be for climate change. If we can get these bacteria up and running, then we will really want to be less reliant on mineral fertilizers in the future. I believe that phosphorus stocks are running low, but if you just do a standard soil analysis test for total phosphorus and organic phosphorus, you'll find that the soils themselves do have a huge reserve, or most soils have a huge reserve of phosphorus in there. And what we really need to do is unlock that reserve that's in there instead of just making more phosphorus fertilizer, mining it, converting it into fertilizer, producing waste gases, using up energy. Why don't we start looking at what's in the soil and try and unlock that? And the way of doing that is either with low molecular weight organic acid, which I talked about a little bit, very tiny bit. I'd like to talk more about it. And also by the microbes as well, because they also have the potential to unlock lots of, I'm just talking about phosphorus here, but they have the potential to unlock that phosphorus from the soil. And if we can unlock what's already in the soil, we don't need to worry about producing, extracting, mining, chemically converting more phosphorus from phosphate rock to a more, let's say, easily available form of phosphorus. I'm just using
Yes.
phosphorus as an example, but
Yeah.
same could be true for sulfur, nitrogen, things like that.
Yes, and it was a very good example because I was just thinking about our soil microbiome course that we are going to launch in March. And one of the examples is how we can unlock some of that soil potential when we do a lot of work around those wonderful bacteria and fungi and soil microbiology in order to achieve sustainability, productivity, and also to save money to farmers. Because if the nutrients are there, you don't need to spend money buying more
Mhm.
nutrients. Alan, it was fantastic. Thank
Thank
you so
you.
much for your time here. Next time I will give you just a full webinar just for yourself.
Right, yeah, okay.
Okay, thank you.
Thank you.
And I will now call Ruth. Hello, Ruth. You're already here with us. So when you're ready, let's go into enzymatic analysis. Let's see
Yes,
what they can do for
hello,
our
Claudia.
soils.
Thank you for the invitation to participate in this interesting webinar. I'm sharing my screen, and also thank you for the presentation. So I hope that you are already seeing the slides. Yes?
Not yet, Ruth.
Well,
Well, again, we tried it before, so it worked. Now it's
that's
coming. It's coming.
now
It's fine. Perfect.
that's fine.
Yes.
So good. So,
Slide two.
good afternoon to all. Since I always never know who is from the on the other side, so I don't know exactly the public. I'll always have some difficulties if I should go to the basics or if I should not go to the basics. But I think that is always important for all of us to remember some basic concepts. And so let's talk about soil organic matter and soil enzymes. When we talk about soil organic matter, we are talking about biological components. So soil organic matter is produced by organisms and it enters on the soil after the decay of these organisms, animals and plants and microorganisms. So the soil organic matter is the source of biomolecules. And when we talk about biomolecules, we can concentrate on the dominant ones that all of us know. So we are talking about carbohydrates, we are talking about proteins, lipids, nucleic acids. And all of these molecules are complex molecules. Uh, all of them have in their chemical composition carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. When we talk about soil fertilization, these are not the only nutrients that we are concerned about, but are the dominant ones that we are that we usually to talk about. So all of these nutrients, all of these elements are present on soil organic matter, and all of them compose simple molecules like that we use called monomers, which are monosaccharides, amino acids, fatty acids, glycerol, and nucleotides. And these monomers form the polymers, so they combine themselves in polymers, which are the most complex molecules that we found in the organic matter. And um, and now let's go to the soil organic matter mineralization. So what happens to these polymers when the soil organic matter is degraded in the soil? In fact, we have the soil enzymes that are responsible for this work, and they carry out enzymatic reactions, enzymatic hydrolysis or enzymatic oxidative hydrolysis, and they start with mineralizing the polymers that we were talking about, and they end up transforming these polymers into monomers, and the monomers are assimilated by the soil microorganisms that are responsible for producing this enzyme, are assimilated by them because they are very relevant for these microorganisms as a source of energy, and also because they are very important for their metabolism and for being transformed again in polymers that will constitute the cells of the microorganisms. So what you see here is that, in fact, the soil organic matter that enters in the soil when the decay of living organisms happens, some of them are already simple compounds, some of them are already monomers that are immediately assimilated by the microorganisms, but other ones are complex organic molecules that need to be mineralized by microorganisms and by their enzymes, and then to become monomers, and then they can be used by assimilated, or they can become available to the other organisms, namely to become available to plants for being uptaken and for being used also to produce again polymers, or to produce again more complex molecules. And when we talk about enzymes, we are talking about specific proteins that have a role. Some of them are catalysts, so they are responsible for activating reactions, catabolic reactions. And these enzymes, these specific proteins, they can be intracellular enzymes, so they can be inside the cells of the microorganisms. They can be integrated in the membranes of these microorganisms, or even associated. They can be extracellular enzymes, but associated to the cells of microorganisms. When the conditions are not favorable in the soil, let's, for example, think about very low temperatures as we have today, what may happen is that these cells are less active, so they are dormant, and all this machinery is not working as it's supposed to be. But also something that may happen is that these microorganisms may also die, and they can start being accumulated. This dead organic matter that is part of the microorganisms can be accumulated in the soil through something that I forgot to mention in my previous slide, which is the entombing effect. So it is the accumulation of organic carbon that makes part of this microbial biomass that accumulates in the soil when the decay of microorganisms also happens, and when this organic matter starts being degraded. Of course, that some of these enzymes are also degraded or can become extracellular enzymes. But most of these microorganisms, what they also do is they synthesize extracellular enzymes that need to be stabilized by the soil, and they can be stabilized in a different way. So they can be complexed with their substrate. They can be complexed with soil organic matter. They can be complexed with clay minerals, or they can be also complexed with complex organic matter molecules. This is, which means that something that I will talk later, that the soil properties are also very important for the stabilization of these soil enzymes. But what is a simple soil enzyme? So it is a protein. We have already talked about that. It is a specific protein that has an active site where it interacts with a specific substrate. So usually they have some level of specificity for the substrate with which these enzymes interact, and they form the complex with their substrates, and they promote the catabolic reaction, giving rise to the monomers of the substrate that they are catabolizing. And this is the Michaelis-Menten kinetic curve that demonstrates how the activity of these simple enzymes happens, which means that when the concentration of the substrate, the velocity of the reactions that I'm describing here above, increases to a certain level, but then it's stabilized because the active sites of the enzymes are saturated. So if we have more substrate, doesn't mean that we are going to have more metabolic activity. And let's now understand why these enzymes are important. So they are involved in just to summarize, in the mineralization of polymers, in the composition in their monomers. And so let's look to a biogeochemical cycle. And again, let's go to the nitrogen cycle, which is one of the cycles that most concern us in the soil. And when we look to the biogeochemical cycle of nitrogen, I will not go to this part, which is related with the fixation of nitrogen with specific microorganisms in the soil. But let's start from the mineralization of organic matter. So when the enzymes start mineralizing organic matter, what they will do is to remove the nitrogen from the organic compounds. And the first step is the production of ammonia. So we have a first step, which is ammonification, which is the transformation of organic nitrogen in ammonia. And this is an oxidation reaction. And you see that the production of ammonia doesn't come only from the organic matter that is in the soil, but it also comes from residues of animals, for example, like urea, or it also comes from the use of fertilizers based in urea. So the application of urea in soil gives the substrate for the ureases, and these ureases will convert urea in ammonia. So we can get ammonia in the soil from two different types of sources. Afterwards, what is going to happen to this ammonia? We have what we call the nitrification process, which is very important because it's the conversion, which is again an oxidation process that transforms the ammonia in nitrates and nitrates, which are the main source of nitrogen to the plant. So we are starting here to have nitrogen compounds that can be used by plants and then can be assimilated by plants and converted in complex organic nitrogen. Afterwards, we have a final step, which is the nitrification. So now we need the absence of oxygen, and we have different types of enzymes that will going to transform the nitrates in nitrogen oxides that comes back to the atmosphere again. What I want to say with this is that when we analyze soil enzymes, we should be careful which type of soil enzymes we are analyzing, because we have soil extracellular enzymes acting on different process of this nitrogen cycle. So which means that we cannot make any conclusions if we don't measure all the enzymes that are at these different steps. So we need to understand, if we use the enzymes to understand how the biogeochemical cycles are running in the soil, we need to measure all the enzymes that are working at the different steps if we want to understand the process and if we want to understand what is happening. Let's just give you some more examples. When we talk about phosphorus, which is a very high important nutrient for the plants. In fact, what you see here is that, okay, we also have phosphomonoesters, which let's simplify the names. We have organic phosphorus in the soil, which comes from the organic matter. And what the phosphatases will do, they will convert this organic phosphorus in phosphate ions, and the phosphate ions are the form of phosphorus that the plants can use immediately. The same happens with sulfur. So we have sulfate esters in the organic matter. So we have organic sulfur in the organic matter, and resulfatases, what they are going to do is to make the catabolism of these molecules, of these organic molecules, and convert them in sulfate ions, which is the form of sulfate that the plants can also use. So we have different soil enzymes that are working in different soil biogeochemical cycles. And if we want to understand, we have a global overview of all of these biogeochemical cycles in the soil, we really need to look to all of these enzymes to interpret what is happening. But anyway, this is not easy. And this is only one of my concerns, is that, or one of my recommendations is that we need to be highly careful with the conclusions and with the recommendations that we make when we analyze all of these biological indicators, chemical indicators in the soil. Just to give you one example of one study that we perform in different Douro vineyards, in 3 plots, in 3 Douro vineyards, where we want to analyze several biological indicators and chemical indicators as well. In the plots of these 3 vineyards, 2 of them were under integrated production mode, and another one was under organic production mode. And what we see here, we have several parameters, but since I only have 15 minutes, I don't want to spend more time. What you see here is that looking to organic matter, but it is in percentage, we are talking about organic matter, not organic carbon. What you see is that the 2 plots of the 2 vineyards under integration production mode, this one and this one, have a high percentage of organic matter when compared with the vineyard in organic production mode. And here are 2 references soils that were collected nearby the vines. The vineyards. If we go to the soil enzymes activity, what we see is that, okay, if we have more organic matter, it makes sense that we have more biological activity in the soil. And the hydrogenases enzymes are enzymes from that are involved in the respiration of soil microorganisms, and this means that they are more biological active. So if we have more organic matter, they are metabolizing the organic matter and they are consuming oxygen, and they are respiring, they are doing their cellular respiration. And this was measured in different times of the year, in the winter, in the early, late spring, and in the winter again. And we see that the biological activity was higher, of course, in the late spring. But when we go to the vineyard under organic production, the biological activities of these enzymes was very low. The metabolic activity of this enzyme and of the microbial community was very low. What makes sense when we have few organic matter. And then if we go to the other 3 enzymes, just showing these ones, you see that if we look to aryl sulfatase activity, it still be higher in at least in one of the farms under integration production mode, and it was higher in February, probably, we don't know, but probably because there was a fertilization, or it was the addition of sulfate organic compounds to the soil. Let's look to the acid phosphatase activity. If you look here, you see that the activity of the acid phosphatase activity are higher in one of the farms under integrated production mode, and in one of the farms, in the farm with organic production mode. And of course that in this case we have more organic matter. So if we have more organic matter, we have more organic phosphorus. But what makes sense, because we have more biological activity, but in this case of acid phosphatase activity, their activity needs to be carefully interpreted, because this enzyme is activated when we have low phosphorus available in the soil. So in this farm, we have few low organic matter, but we also have low availability of phosphorus. And when we go to the nitrogen mineralization, only one of the steps of the organic nitrogen mineralization, we see here that once again we have in this case more activity, more nitrogen mineralization mainly in the farm under organic production mode, in integrated production mode, sorry, but once again a lower activity in the farm under organic production mode, once again because the organic carbon, the organic matter was lower in this farm when compared with the others. My issue here is that we start doing this work aimed in analyzing the impact of the practice in the soil, but what we realized is that not only the practice that is influencing the activity of the soil enzymes, but it was mainly probably the organic matter that was influencing the activity of the soil enzymes in the soil. So just to summarize and to keep it as simple as possible, enzymes are fundamentally of microbial origin, so they are released by archaea, bacteria, and fungi to the soil to help the microorganisms to mineralize organic matter and to get the nutrients that they need to assimilate and for using as energy and as source of their polymers, of their structural molecules. But there are also soil enzymes that are released by plants and animals, although they are not so representative in the soil. These enzymes mediate catabolic organic components, so they are correlated with organic matter, they are correlated with soil physical properties, biomass of microbial community, and with many other factors. So it's highly recommendable that we evaluate a battery of enzymes and try to include enzymes that are involved in all the nutrient cycles, and also enzymes that are involved in the different steps of these nutrient geochemical cycles. We should use as much as possible standard protocols to allow us to keep the possibility to compare the results between studies, or if we are not able to use standard protocols, at least to use well-established protocols. And also, we should interpret the results of soil enzymes and also of all the soil microbial parameters very carefully, because they are highly influenced by many, many variables. So we can never take direct conclusions related with soil management practices because they are influenced by soil properties, climatic conditions, and so on, soil types. So there are many issues, many variables influencing these parameters, which means that I'm talking about soil enzymes, but they should always be combined with other parameters. Like, for example, we are discussing here today metagenomics, the test, for example, the respiration test, or other tests that allow us to evaluate soil respiration, as the one that Alan presented, degradation of organic matter tests. So there are many other tests that then should be performed together to help us to interpret soil health. And also, one of my recommendations is that we should be highly careful when we talk about soil health and soil fertility, because they are really different aspects. And when we talk about fertility, we are talking from a different thing than when we are talking about soil health. So thank you for your attention. I hope that I didn't surpass the time. I was very stressed with that.
Aren't we all, Ruth? Aren't we all? It's all very short, but you were brilliant, and I think
yeah,
you've just finished in a very good way. The
mhm,
devil is in the details, and Alan
mhm,
just posed here a question. And I'll go back to that, and those are the details. But just to point that here at Food for Sustainability, when we do our soil analysis, this is something that we take with extreme care, is to fully understand the context and the where we are making the recommendations, very close relationships with the farmer to understand, the management practice, what's going on, the endophyclimatic and the history of the farm, because
mhm,
then
mhm.
that can change the entire analysis. And actually, Alan says that in the Douro Valley, years of use of copper sulfate as an antifungal agent has contaminated many of the soils with copper. This copper can inhibit enzymes. Are there any consequences for the analysis of the enzymes because of this copper?
Yeah, it depends what we talk about high levels of copper. So it depends on the vines. It depends on the vines are managed because you know that soil is mobilized. Sometimes the copper goes when the soil is mobilized. The copper goes to greater depths where we don't have biological activity. There are many variables. So and the levels of copper in some areas, what we are seeing is that they are not so high as some studies report, because what happens is, this is another aspect, is that sometimes conclusions are made based on very few samples, and some of the European studies that are being reported sometimes are based on very few samples, and they report very high levels of copper, and then the copper is not so high as expected. And also more important than the total concentration of copper, and I believe that Alan agrees with me, more important than the total concentration of copper is the available concentration of copper. In the most cases, in at least in several sites where we have been studied, sometimes the concentration of total copper are relatively high, but then it's not available, so is not causing effects on soil biota. So all of this, it's another aspect that we need to take into account. Yes, yeah,
Okay.
that's true.
Thank you for that clarification. Ruth, while the time runs very fast, and while I call Joana to the stage, just a clarification. So these seem very interesting analysis. I know that you've been working with them for a while. Um, do you conduct this analysis at the lab as an open source, so for everyone, or is it exclusive for research? And can we have a range of values of how much would this analysis cost?
Well, if someone from my research team is hearing me, they will kill me tomorrow. But the question is that we run this more or less in a routine base for research. We can do as a service outside, and of course that we can provide a value for this analysis, because we do it for many years, at
Mhm.
least since my PhD, that I'm doing this type of analysis, and now I'm not going to the lab anymore, unfortunately, but my research team do it. Yeah.
Okay, so and
Mhm.
probably they will kill you because they are very busy
Yeah,
already
more
with
work.
the loads of work
Yeah,
that
yeah,
they
yeah,
have,
yeah,
and
yeah.
they will probably
Mhm, mhm,
refrain from
mhm, mhm,
more.
mhm,
Thank you so much, Ruth.
mhm.
Ruth, you have a couple of questions in the chat while I move on to Joana, just because of time. Do you think you can have some time just to answer Fadua, who has two
Yeah,
questions for you?
I can write. Yeah, I can
Yeah,
do
yeah,
it. Yeah,
yeah,
yeah.
that's
Mhm.
brilliant. Thank you. Hello, Joanna. Now we're going to hear about metabar coding. We still have few time. Joanna, hello. Are you there?
Hi. Yeah, I'm
Hi.
here. Can you
Y-
hear me?
I can hear you.
Good.
Cannot see you, but I can hear you.
You can't see me?
No, no, but it's probably there you
Ah,
are. Okay, fantastic.
Okay.
So you can just
So
share
I'll
your
share,
screen.
yeah, a second. Can you see it already?
Yes, all good from this end.
Okay. So I'll start just to keep everyone engaged. I know it's a bit after 5, but let's try to run this at least out of curiosity. So today I'm going to speak to you about soil microbiome analysis on a perspective of from DNA sequencing. Just sorry. Okay, so just to let you know, I will talk about briefly about using the microbiome as an indicator of soil health. I will give a very brief, very brief overview of microbiome analysis methods, some of which we have already mentioned here with the presentations of Alan and Ruth. And then we'll go to the details of microbial profiling using NGS. So I'll go over the workflow and I'll just kind of let you know how we move from data that is generated using this technology to actual insights on the microbiome. So first off, to give you an overview of the questions that we want to answer when we are talking about using the microbiome as an indicator of soil health, I always highlight these 5 questions. So the first one is related to microbial biodiversity. So what are the organisms that are already present in soils and how are they interacting with each other and with plants in this specific context of plant production? Then how will soils function, react to agricultural practices? So the question on functionality of these soils. And then a question related to resilience of the soils, which goes a bit in the direction of the question that was here in the chat a bit earlier. How do we achieve robust soils in different conditions? The example being climate change, for instance. Then we also look at the microbiome as an indicator of the nutrition status. How are the practices that are being used impacting the nutrient cycling? And what is the general state of soil health? So considering all this and in considering the potential presence of plant pathogens, what is the general state of soil health? So to answer these questions, we can use several methods. Some of them have been already mentioned here, but I would like to divide these 2 methods in culture-dependent methods and culture-independent methods. The culture-dependent methods, as the name says, rely on growing these microbes in the lab. Okay, so using specific growth media and then identifying them through a microscopy. For example, for fungi, we can identify fungi using a microscope, or to identify bacterial colony morphology, for example. So this relies on a specific settings that only allow you to go so far in identification of microbes. First of all, you need to be able to grow them, and we know that only around 1% of the microbes that exist in soil are actually culturable. So the vast majority of the organisms cannot be grown in the lab, and so that will limit the identification that we are able to do using these methods. And of course, there's only so far you can go on direct observation. You probably will need to rely on a molecular identification strategy such as Sanger sequencing to reach the genus level or the species level. Then on the contrary, we have culture independent methods, and these we can think about all the methods that do not require the microbes to be grown in the lab. And this includes, for example, the functional profiling using the analysis of enzyme activity, as Ruth mentioned in the previous presentation. But it also includes the molecular profiling, and this molecular profiling can be performed using several different compounds that exist inside the cells or at the surface of the cells, okay? So we can talk about extracting DNA, and we can talk about extracting lipids that exist on the microbe membrane, okay? But for the sake of this short webinar, I will be focusing on the DNA molecular profiling, okay? So the most common technology that we use currently is the microbial profiling using next generation sequencing. Okay, this is a brief overview of the workflow. It starts with sample collection, of course, so it involves a very specific protocol for collecting the samples and transporting them to the lab. And then in the lab, we do DNA extraction and sequencing, and a lot of data analysis and result interpretation. So this technology allows to generate a ton of data that will need to be translated into actual actionable insights in the field. So going over what we do in the lab, the DNA extraction is, I would say, one of the most critical steps. As you know, soil is a tough matrix, so it includes a lot of inhibitors, and it also includes a lot of organisms that are hard to lyse, that have tough cell walls, and so it will be hard to release the DNA that is inside the cell. And so for this, we rely on a mechanical and chemical cell lysis, okay? That this is going to allow us to effectively burst all of these cells and get the DNA out of the cells. And this extraction step is also it includes also a step of inhibitor removal, okay? Because these soil samples are rich in humic acids that are going to inhibit all downstream applications, from PCR to real-time PCR to sequencing. So if the sample, if the DNA is dirty, as we call it, it is not going to yield good results, or even any results sometimes. So this process of extracting DNA is critical. We use at the lab an extraction robot, so it's a semi-automated extraction. This allows us to kind of standardize the yield and the quality of the DNA that we get, even from more challenging samples. And we also have this step of quality control, in which we check concentration, purity, and integrity of this DNA before moving forward in the analysis. Okay, and how does this actually work from DNA to sequences? So this starts with a PCR amplification, and this PCR amplification targets conserved regions of the microbe's genome. So in this case, we are talking mostly about bacteria and fungi. For bacteria, as Claudia mentioned, we use the 16S rRNA gene, and for the fungi, we use the ITS. And these are conserved regions that are going to exist in all of these organisms. So 16S is going to exist in all bacteria and archaea, and ITS is going to exist in all fungi and also all mycetes. So we are targeting these conserved regions of the genome, but these conservative regions, they actually flank variable and informative regions. So we are going to target that big region, but the goal is to look inside that region and find differences that we can actually use to differentiate the organisms that are present. And we do that by a PCR amplification. After PCR amplification, we have a library prep step, and that means that each sample will have total DNA that will be amplified for 16S and for ITS, okay? And for each sample, we will do a barcode and adapter ligation. This will allow us to run multiple samples, more than 100 samples at the same time, and also bind this adapter that will allow ligation to the physical support in which sequencing happens, which is a flow cell. So these adapters are synthetic DNA fragments that are added to our PCR products, and then they are going to be able to bind to the flow cell, which is where sequencing actually happens. How does this happen? Each library is bound to the flow cell, and then they are going to be amplified again, so multiplied again through bridge PCR to create clusters of sequences that are bound to the flow cell. Then what happens is that after these clusters are bound, new sequences based on the template sequence are going to be produced, and each time a nucleotide is added, the equipment takes a snapshot, okay, takes a picture. And on this picture, we can see different colors. So each color will represent a nucleotide, a letter on the sequence. And that generates millions of reads in parallel that will then have to be translated into actual information. How does that work? That's where data analysis begins. So the first step is to do this initial processing of all of these millions of sequences that have been generated in the sequencer. This means trimming for the appropriate size and quality filtering of the sequences to make sure that we are only continuing the analysis with high quality sequences, okay, of course, to increase the confidence that we have in the results. Then what happens is that we will need to cluster these sequences. So we need to group similar sequences, because similar sequences will most likely belong to the same organism, or at least to very closely related organisms, okay? So we group the sequences into these similar sequences groups. And then after we have this clustering, what we do is matching against the reference DNA sequence database to achieve a taxonomic ID. Okay, so this is taxonomic alignment. So we will have public databases of 16S sequences, of ITS sequences, and we will match our sequences that we get in the from the sample with those databases. And that is going to tell us, okay, so this sequence belongs to Pseudomonas aeruginosa, this sequence belongs to Bacillus subtilis, and so on and so on. And the number of sequences that we find belonging to organism A, B, or C will then generate a composition of the microbial community. So where we retain the relative abundance of each species based on the relative abundance of sequences. And this leads to a very boring table like this, okay, which has a lot of information that actually doesn't convey any specific information to the producer, but this is the first output of our analysis. So it is a list of species and a list, sorry, of its relative abundance. Okay, so of this output, we actually need to make something that is actionable and understandable to the producers. How do we do that? So here enters the again questioning the data. So first thing that we try to answer is on taxa diversity, so based on number of identification, which is how many different organisms were found. Okay, so did I find 2 species or did I find 1,000 species? Okay, how many? What are the major groups of organisms that are found in this sample? Okay, is my sample composed only of bacteria, or do I have archaea and also fungi on this sample? So diversity, an overview of microbial diversity. And then drilling down on that, we have relative abundance. So based on the read number, what are the most abundant organisms? Is the most abundant organism a plant pathogen? Probably I'll have an issue with that, okay? Or is the most abundant organism plant growth promoting rhizobacteria? Okay, or I don't have a most abundant organism and the microbiome composition is even. So all organisms are present in a more balanced way. Okay, we can do that by adding this type of visualization, which helps in understanding what is the microbiome. And then moving on a bit on this, and of course related to the species that were identified, we can do diversity measurements. So is my sample high or low diversity? So within the sample, we can calculate alpha diversity. Okay, it's of course diversity is always based on the number of species and the evenness of the species that were found, and we can also understand if how is the diversity between samples. So is sample A more diverse than sample B? For example, if we are comparing practices, do I have more diversity in a sample coming from taking Ruth's example, organic production, or do I have a more diverse microbiome on samples coming from my reference soil? And finally, to add a layer of information to that, we also do functional diversity assessment based on ecological and metabolic functions found for those organisms. Okay, so this is not a prediction based on functional genes, okay, but based on what we already know, based on the literature and existing databases, that these microorganisms that were identified are able to do. So did I find a nitrogen fixing bacteria? Did I find a plant pathogen? Did I find, I don't know, as you can see here, for example, in this graph, you see nitrogen fixation, nitrate respiration, nitrate reduction. So this is something that we add to the information of what was found, and we do that by matching taxonomy assignment against the functional database. We use public databases like FAPROTAX, FunGuild, and Tax4Fun. We also use a proprietary database from Biomakers, which is the B-Crop database, and we also have our own in-house built soil microbiome database, which combines all of these functions, but on a more crop-specific approach. So we will have, okay, I found, I don't know, a Champomonius perforans. I want to know if that organism is a plant pathogen, and if it is a plant pathogen, what are the what is its host range, for example, and how can I use other organisms to control this? So our database will let us know and enrich our results with this type of information. So to conclude, since it's almost 5:30, to conclude, what I would like to say about this technology and about using this approach to characterize soil microbiome is that the key insights is that we have a biodiversity assessment, we can detect rare or previously unknown organisms, and we can do at some point a functional prediction of what the organisms in this soil are able to produce, are able to perform, sorry. And the key advantages of this method compared to other microbiome analysis methods are that it is high throughput. It allows for testing a lot of samples at the same time. It is culture independent. It has a high accuracy and resolution, and it's also a very cost and time efficient method to make this big overview of the microorganisms that exist in the soil. So thank you. Please.
Thank you so much, Joana. It's always a pleasure to hear you. You left me 1 minute. Please don't go away. Don't go away. You have a few questions in the chat. If you can just look at them. But while I open the discussion to all the speakers, Joana, Alan, and Ruth, if you are into this topic, let me just remind you that we will have a soil microbiome course analysis, really to help you understand how you can interpret and can make decisions based on the outcomes of the analysis that Joana was presented. Joana is one of the speakers at this course. So if you are interested, keep an eye on this edition. It's going to be the third edition. It's going to happen in March. Also, as promised, and still on time, here is the QR code for your assessment. You can perform your assessment while we keep with the conversation with our speakers. I will also ask the team to place this link in the chat, just for those that find it difficult to use the QR code. And while you concentrate in the question, but at the same time hearing about all this expertise and knowledge that our speakers bring to us. Joana, I will start with you. There is this question about matter barcoding, death or living microorganisms.
Yeah.
Please go ahead.
So DNA-based approaches will not be able to distinguish between live or dead microbes. We are looking at DNA, and this DNA can come from cells that were active in the soil or not. It can even come from fragments of DNA. What we know is that DNA tends to degrade, so most of the results will be coming from cells that were alive, at least until very recently. Okay, so but no, short answer is no, it cannot. It cannot distinguish between live and dead cells. If we want to distinguish between live and dead cells, we would have to use, for example, a technique based on RNA, for example. So transcriptomics will allow us to see exactly what are the organisms that were expressing genes at the time of the collection. So transcriptomics would be a way to go.
And about fungi and spores, can we distinguish those two?
Yeah, same.
Through metabarcoding?
Yeah, no, same answer. So for bacteria and for fungi, the answer is the same. Of course, that this means that, and going a bit back to what we were saying in the previous presentations, is that it is actually important to combine all of these types of analysis to get a full understanding of what's happening in the soil. So this multi-approach on soil health is the best approach, in my opinion.
Okay, thank you, Joana. And then one final question to you all. I think you have shown here that you are extremely knowledgeable about these techniques and also each other's techniques. Food for Sustainability and A2, they take a more commercial approach. They want to be closer and ensure that this democratization of knowledge in terms of how we measure soil microbiome. Ruth has explained also that they can proceed the samples, enzymatic analysis, but the core is actually research. But from your work, can you share some real examples, case studies, such as papers, farm transitions, profitability data that can show how this soil analysis, matter barcode, Henning test, and the enzymatic activity are actually driving farmers towards more sustainable, resilient, and in the end, delivering high profitability at the farm level?
Yeah.
Can we relate this to farmers? Go ahead, Joana.
Yeah, definitely we can. And what we have seen since the lab began to function, which was not that long ago. So we started the lab. The lab was inaugurated in October of 23, and we actually only started to perform analysis in May of 2024. So we have 1 year and a half of activity, and what we see is that the number of farmers that reaches out to us to understand more about the microbiome and to test on their own parcels this kind of technology with their own settings is growing every month. So we have new customers coming in every month to test their soils. I would say, for example, that one of the best examples that we have is a producer of aromatic plants that, based on the results, on the initial results of microbiome and physical chemical analysis, conducted a baseline study, and that baseline study identified several actions that could be taken from a microbial enrichment point of view. And now, 1 year and something later, what we are seeing is that after those actions were taken, introduction of microorganisms, changing some of the practices, what we see is that for that specific producer, they were able to harvest once more when compared to the previous practices. Yeah. So, and that is profit for the farmer. So, in terms of acceptance from the farmers, what we are seeing actually is very, very good.
Yeah, that's an interesting case. That's when the bottom lines of farmers actually improves. Thank you so much, Joana. And I know you've been also around and with farmers for a while. Is there anything that you have to share with farmers at a more practical level?
Is my microphone still on? Yes, can you hear me?
Yes.
Okay,
yes,
that's fine. Really, what we've taken is that the people or the farmers who were already in regenerative agriculture, so even before Haney tests became popular, they were already using like cover crops and things like that. Their results actually show that the soils are actually do have more living microorganisms in there by the amount of respiration that we see, the amount of water extractable organic carbon, for instance. So we actually see that using these regenerative agricultural practices, they can increase the amount of cycling of nutrients that happens in the soil. So they increase the microorganisms, and then the bills that they have to pay for fertilizers reduces, which once again, like Claudia said, it's influencing the bottom line of the farmers. If they don't have to constantly pay so many €1,000 for fertilizers, that's all money that's saved and in their pocket. Why not get the microorganisms to do it for free? So that's what we are kind of seeing, and that's why more and more people, as Joana said, are moving towards like regenerative agriculture, looking at the microbiome as well, to see how money can be saved by not having to constantly buy products from salespeople.
And it's important to make that case that we actually have workers with us that can do the job, and we don't need to outsource through synthetic inputs. Thank you so much, Alan. Ruth, you are with your camera off, but I believe you're there. I know that you've been working a lot, lots of projects, national and international. So eventually have something also to share with our audience.
I'm sorry, Claudia. I didn't saw that I have the camera off, but I'm always here hearing you. Well, I'm scientist, so which means that sometimes I'm not so positive as my presenting colleagues. What I could say to you is that that farmers are indeed more aware of the importance of soil biota and the importance of the role of soil biota in soils, and they in fact are looking for all of us to understand what is happening and how to promote the activity of the soil biota, and this is very, very good. What I would like to say is that there are lots of things that we still don't know, and that we need to do more research. And I invite you to follow, for example, the Living Soil Project, which is an European project that is creating living labs. And at this moment, we have 67 experimental sites in 5 European countries, where we are implementing a soil monitoring framework precisely to understand how soil biological and chemical indicators change when we change the practices. And this will be done throughout the time, and we hope that it will continue after the project ends. And I think that only with long-time data and a systematic monitoring of all of this, we can make some conclusions. And even though it's very difficult, because there are lots of things always changing. So that's
Which
all.
is good also,
Of course,
because
that is good.
there's still the
Yeah,
role of science.
definitely.
Yeah, that's
Yeah,
still, we
definitely.
still have a lot.
And we
So we
need
have,
to start, and we need to
yeah,
start, and we need
exactly.
to start working with farmers, doing the best as we know, because we are all of us are learning together. So that's
Yes,
the positive
that's true.
point. Yeah.
Yes, and you mentioned an interesting point. We should feature also the living labs, and particularly
Yeah.
those where Portugal
Yeah.
is leading, and showcase them here as sharing
And also, yes.
the
And
knowledge.
Food for Sustainability also have test farms and so on, so which are
Yeah.
also have more or less the same role. So you want to demonstrate and to show. So that's what we have to do together, all of us together. Yeah, that's the important.
Yeah, exactly. Ruth, thank you so much. We won't have time for that today. I'm sorry. It was really a pleasure to be here. I failed to finish this webinar sharp at the hour, but it was a pleasure for me to be here. Thank you so much to our great speakers, and also to this audience and staying this longer. It was great. Just to remind you that we will be back next week. We will actually go into some of the issues that were already highlighted here. We will look how inoculation actually happen, and how this inoculation can actually be an acceleration process for more living and healthier and functional soil. So we count with you on that side next week. It was a great to moderate this webinar. Thank you again for being here. See you next week. Bye bye.
Congratulations to
Thank
Food
you.
for
Bye, everyone.
Sustainability as well. Bye bye.
Bye.
Thank you, Ruth. Bye bye.
Bye, everyone.
Bye.
Bye.